related to promotion to full professor consideration. So it's a real pleasure to be able to introduce Eric. He is a world expert on the biology of a parasite called Toxoplasma gondii. And if any of you in the room look one person to the right or one person to the left of you, probably about one of the three of you have antibodies for this particular parasite. So indeed, it's a very common, successful parasite. So just a little bit of background about Dr. Denkers. His formal education began in the Pacific Northwest. He earned his bachelor's degree in 1982 at the University of Washington. He worked there for a couple of years at the Cancer Center, the Fred Hutchinson Cancer Center, before heading off to the University of Wisconsin-Madison, where he earned his PhD in 1990. His PhD work was also on a parasite, in this particular case, Trichinella spiralis. And after finishing up in Madison, Dr. Denkers went on to join a very distinguished group of immunoparasitologists associated with the intramural program at NIH, working in particular with Dr. Alan Scher, one of the world's experts on immunoparasitology. And he was a postdoctoral colleague and then a senior colleague there from 2000 to 2005. And then, right thereafter, he joined the faculty at Cornell University in the School of Veterinary Medicine and was promoted through the ranks, assistant, associate, and full prof at Cornell. And then, to our great fortune, he decided that he had enough of Ithaca's gloomy weather and decided to head for bright and sunny skies. And Dr. Denkers joined our faculty in 2016. Like I said, he is an expert on Toxoplasma immunobiology. He's published around 100 papers on that topic. He's been very successful in getting funding to pursue this work. He's had a couple of long-standing runs with NIH to investigate signal transduction pathways in Toxoplasma, and also with the interactions between Toxoplasma and neutrophils. So without further ado, I'm going to turn this over to Dr. Denkers, and he's going to tell you about Toxoplasma gondii, harnessing immunity for a successful host-parasite relationship. Eric.
Thank you, Sam. That nice introduction. So, let me just things up here. Okay, yeah, so thank you for that introduction. Thank you for coming to my talk. And so, I'm going to talk about what I've been doing, working on Toxoplasma, both before I came here and in the 2 years since I've been here. So beginning with this image here. So this is an infected macrophage here. And these are parasites inside the cell. So this is Toxoplasma gondii. It's an intracellular parasite. It replicates inside these cells. And so really what I'm, in general terms, interested in is the biology between the parasite and the host-parasite interaction. I wanted to actually, I thought this might be a good place to begin. And that's kind of thinking a little bit about evolution. So I'm an immunologist, and I actually don't think as much as I should about evolution. I don't think many immunologists do. But this paper came out back in 2004, written by an immunologist, a well-known immunologist. And it's a paper kind of taking into consideration the process of evolution in the host pathogen interaction. And it was really, to me, it's an amazing paper. It was a real sort of eye-opener. And so this is the abstract of the paper. It's also really a beautifully written paper. But the point of this paper, if I could try and summarize it, is that we, meaning me, meaning other immunologists, tend to think that in terms of evolution, we are continually evolving our immune system towards what Stephen Hedrick refers to as the optimal solution, which is the solution that we get to, the state where we get to, where we are completely resistant to infections. We develop our immune system, we no longer have to deal with infections. And so the point of this paper is that that is a completely fallacious point of view, and that as the immune system evolves, as it undergoes variation, really all we're doing is we're selecting for variants in the pathogens that infect us. And so for every evolutionary move we make, there's a counter move by the pathogens that infect us. And so really what we're doing, in some sense, is really continually selecting for smarter and smarter pathogens that can infect us. And that was really the point of this paper. And so it's really, as I said, it's an amazing paper, really nicely written. There's some really nice sentences in here. I think everyone should read this. So he ends off by saying that by selecting for ever more devious parasites, I like this part, the immune system is the cause of its own necessity, which kind of makes it, it's kind of like a Zen koan or something. I really like it. And so, of course, this refers to the Red Queen hypothesis, really. And so the idea comes from, the Red Queen concept comes from Lewis Carroll and Through the Looking Glass. And so there's one scene in that book where Alice, here's Alice, it's a beautifully illustrated book, by the way. This is an original illustration from the book. And so Alice meets up with the Red Queen in the fantasy land Through the Looking Glass, and they both take off running at a huge, very fast. They're running and running. They eventually stop. They're out of breath. They're completely tired, and they look around, and they haven't moved. They're in the exact same place. And so the Red Queen here, if I can read it, says, here you see it takes all the running you can do to keep in the same place. And that's really the situation we find ourselves in our encounters with the pathogens that infect us. No matter how much we try and evolve to combat these infections, there's always a counter sort of, and we never escape the problem of infection. And so what I'm going to try and show you today, what I hope to show you is in my work on toxoplasma, really this principle in action. And I wanted to start out, or actually not quite start out, it's a second slide, but I just wanted to show you a movie of toxoplasma. It's a fascinating parasite. It's an intracellular parasite. Here it is right here. This is a host cell. This is a fibroblast. And so what you can see here is that the parasite actively invades. It penetrates the host cell. It has invasion machinery that enables it to enter the host cell. So it enters the host cell, and inside the host cell it creates a vacuole that we call a parasitophorous vacuole. And so within the parasitophorous vacuole, the parasite lives out its cycle in the cell. It replicates. Eventually the host cell lyses. And so I'll be talking about what's going on during this invasion event in the second part of my talk. There's some really interesting new things that we've discovered about what toxoplasma is doing as it's diving into the host cell. So here's some nice EM images. These were not taken by me. This is by a parasitologist, J.P. Doobie, very well-known parasitologist who works at the USDA. And so he has these really very, very nice images of toxoplasma. Here's the parasite right here. Just to re-emphasize that the parasite actively invades the cell. This is the host cell here. It's kind of pulling, it's diving its way into the host cell. Once inside the host cell, it lives within a vacuole. As I said, this is called the parasitophorous vacuole. And so this form of the parasite is called the tachyzoite. It's a replicative form of the parasite. Toxoplasma is a member of the apicomplexa, and apicomplexa include the parasites that cause malaria, so it's very closely related to malaria. The malaria parasite has a very similar structure. And what I want to point out to you is that these parasites are called apicomplexans because their apical regions have a collection of organelles that are intimately tied to invasion and survival in the host. And in particular, there are one class of organelles called rope trees, or these club-shaped organelles, that are located at the apical region of the parasite. These rope trees contain a collection of, I believe it's somewhere on the order of 200 proteins. And these proteins are released from the rope trees as the parasite invades. And they're known to be involved in creation of this specialized vacuole. They're also involved in influencing the host biology during infection. And I'll be talking about that a bit later in my talk. So this is the intracellular niche where the parasite lives. So this is a single parasite here, but this parasite will replicate. As it replicates, the vacuole gets bigger and bigger. Eventually the host cell lyses and free parasites are released. So I want to go over the life cycle of the parasite. And so that's illustrated here. So there are two phases of the parasite infection. So there's an acute infection and the chronic phase of infection. The acute phase of infection is characterized by invasion of host cells. I already showed you this image here. And so during the acute infection, the parasite invades cells, it replicates in these cells. Eventually the cells lyse and release free parasites. And if you're suffering from an acute infection, you might feel like you had flu, you might feel like you had a mild case of the flu. But in general, it's not something that you would go to the doctor about. You would just kind of feel a little bit off for a few days, and eventually you would get better. And as you get better, what's happening is the parasite is not being eliminated, it's actually going to tissues of the central nervous system and the skeletal muscle. And in these tissues, the parasite forms dormant cysts. So this is a cyst right here. I don't know if you can make it out, but each of the dark spots would be a parasite nucleus. So these cysts contain upwards of thousands of individual parasites, and they're in a quiescent state. In this state, the parasite is referred to as the bradyzoid. This is a section through a mouse brain, and so what you can see, or actually what you fail to see, in a typical infected host is any kind of inflammatory reaction surrounding this cyst. So this cyst is completely quiescent. It's just kind of sitting in the brain. It's not causing any overt inflammation. During this phase of the infection, the parasite is asymptomatic. So this is a very common infection in humans, as Sam mentioned. So I can guarantee you that several of you are infected with toxoplasma right now as we speak. And so when I say that, what I mean is that you have cysts in your muscle tissue and your skeletal muscle, and they're just kind of sitting there in dormant form. So the parasite has a definitive host, and the definitive host are cats, members of the cat family. Rodents are a large reservoir of infection in nature, and so if a rodent is chronically infected and if it is preyed upon by a cat, within the intestine of the cat, the parasites emerge from these cysts and they undergo sexual reproduction. So the cats can release infectious oocysts, and the oocysts are released in the feces of cats. And so the oocysts can be present in the environment, and the oocysts are very, very long-lived. They can live for months at a time in the environment. So anywhere that cats like to defecate, like in vegetable gardens or sandboxes, these would be potential hotspots for infection. This is where you can become inadvertently infected. And the parasite is transmitted by accidental ingestion of oocysts in these areas. So if you accidentally ingest oocysts, the parasite gets back into the intestine, and then it goes back into this acute phase, and then so you have completion of the life cycle. Okay? So the parasite can infects humans, it infects basically any kind of vertebrate. It's a very promiscuous parasite. That's very unusual for a parasite. Most parasites are very host-specific. Toxoplasma is unusual. It can infect almost any kind of host. It infects mammals, it infects birds. So it has a large host range, an uncommonly large host range. One of the things that contributes to the success of toxoplasma, probably the major thing actually, is that this parasite can actually be transmitted by direct carnivorism. So you can become infected by eating meat that contains these cysts. And this is probably the most common way of getting infected, is through ingestion of undercooked meat. So things like undercooked pork and lamb are common sources of infection. So you have to cook your meat well, I guess, if you don't want to get infected with toxa. And this is interesting. Again, this is really something that really contributes to the success of the parasite, and it's really kind of a unique aspect of the parasite. And this is actually thought to be a new adaptation of the parasite. So the latest evidence is that the ability of the parasite to transmit itself directly from host to host emerged only about 100,000 years ago. So really recently, and it's thought to have emerged as a result of crosses in different strains of parasites that essentially freed it from the necessity of going through the cat. Okay? So this parasite does not actually need its definitive host to transmit itself from host to host. If anyone wants to jump in with questions as I go through this, by the way, feel free to. Okay, so I just wanted to emphasize here, it's an incredibly successful parasite. It infects all kinds of different hosts, including humans. So the estimates are that somewhere between 10 and 30% of the human population are chronically infected with toxoplasma. Right? So we're talking about billions of people that are chronically infected with toxoplasma. And not only that, but don't forget that toxoplasma infects all kinds of other hosts. It will infect cats, dogs, mice, rats, rabbits, whatever. So we're talking about an incredibly successful parasite. And I always say it's really the most successful parasite that we know about, the most successful parasite on Earth. And again, the vast majority of us live out our lives without ever knowing that we were infected with this parasite. Usually it's asymptomatic, but there are situations where it becomes a really serious and life-threatening disease. And so I'm showing you some prominent examples here. So it's an important infection in AIDS patients. Toxoplasma is an opportunistic infection. And so that's kind of illustrated here. So if someone is infected with toxoplasma, if they're chronically infected with toxoplasma, so they have cysts in the brains and the muscle tissue, if that person subsequently becomes infected with HIV, as their immune system becomes depressed, these people lose their ability to control the parasite, to maintain it in its quiescent form. And what happens is the parasites undergo a process called reactivation, where bradyzoites within the cysts transform to tachyzoites, which is the proliferative form of the parasite. And the tachyzoites invade cells, they lyse cells, and they cause a lot of tissue damage. And that is what is shown here. And this is a brain scan of an AIDS patient, and right here is a large area of necrosis caused by toxoplasma. So you can see this is kind of like a really kind of serious thing to deal with. And this is a lethal infection if it's not appropriately controlled. There are drugs that control it, but if you don't get the drugs, if you don't get the right drugs, you will die from this parasite. The other situation where we see an impact on human disease, human health and human disease, is during congenital infection. So if someone is pregnant, if a woman is pregnant and she becomes infected with toxoplasma, the parasite can cross the placenta and infect the fetus. Once it infects the fetus does not have a developed immune system and has no way of controlling the parasite, and the parasite can cause a lot of damage to the fetus. It can cause death, it can cause things like blindness and retardation. This is another manifestation of congenital infection where the newborn child has hydrocephaly. And that's caused by parasites in the brain replicating in an uncontrolled manner. So again, a very serious sort of consequence of infection. And the other case where we see an impact on, in this case in animal populations, is in otters in Northern California. And this is kind of an interesting story. It's actually a pretty new story. We don't completely understand it yet. But it turns out that the otter population in Northern California is undergoing a lot of mortality due to toxoplasma infection. So there's something about the strain of toxoplasma in this particular geographic region that is causing infections in otters, and it's killing these otters. And the way it's believed these otters are becoming infected is that oocysts are believed to be being washed into the sea through agricultural runoff. And then the oocysts, based on laboratory experiments, the oocysts are people think that they're adhering to kelp. They're adhering to biofilms on kelp. Then snails come in and they graze on the kelp, and the snails become infected, and then the otters become infected, and then they die. So for the most part, toxoplasma is asymptomatic. We live with it in the vast majority of cases, but there are these situations where it's a really serious infection. So I kind of think of toxoplasma and the immune response in these terms, that there's a real balance between the host response, the host immune response, and toxoplasma. And so this is kind of the way the balance works. So in some sense, the parasite needs to be recognized by the host immune system. And you can see that because if there's an inappropriately weak immune response, the host cannot control the parasite replicates in an uncontrolled manner, and it will quickly overcome the host and kill the host. And you can see that in experimentally infected mice. If you inactivate certain genes involved in host defense, these mice are completely unable to control the infection, and they die within a few days of the infection. So that's bad for the host, it's bad for the mouse, but it's also bad for the parasite, because as any parasitologist will tell you, parasites want to keep their hosts alive long enough so that they can transmit themselves to another host. So the parasite, it wants to avoid an inappropriately weak immune response. It needs to be recognized by the host immune response because it does not want to kill the host. If you go up to the other extreme, if the host makes a very strong immune response, well, that's obviously going to be bad for the parasite because the parasite may be eliminated. It wants to avoid elimination, it wants to stay in the host. It's also actually bad for the host because in certain situations when you have an overly strong immune response, this can result in immune-mediated damage that can actually be lethal to the host. So both the parasite and the host want to avoid these two extremes. And somewhere in between these extremes, you have successful coexistence of the parasite and the host. And so what I always say is that because toxoplasma is so widespread, it's so successful, billions of people are infected, I don't know how many animals are infected with it, but that's really telling you that this parasite is really good at getting to this appropriate balance, this appropriate sort of midpoint between these two extremes. I want to tell you a little bit about the immune response to toxoplasma now, and I'm distilling this to really what you need to know for the data that I'll be showing you today. And so the immune response to toxoplasma goes something like this. So here you have parasites up here. So early during infection, parasites interact with cells of innate immunity. Dendritic cells are an important cell type in the immune response. And dendritic cells respond to infection through receptors like the toll-like receptors, innate immune receptors that recognize pathogens. And as a result of this recognition event, these cells, dendritic cells, make cytokine interleukin-12. Interleukin-12 is really an important cytokine for toxoplasma infection. What interleukin-12 does is it stimulates T cells to become what we call Th1 cells, a subset of CD4 cells. Th1 cells are known for their ability to make this cytokine here, interferon gamma. And interferon gamma, like IL-12, is really important in the ability to survive infection with toxoplasma. And the reason interferon gamma is so important is because it can act on macrophages and cause macrophages to become activated. Once macrophages are activated, they have the ability to kill parasites. So activated macrophages are kind of the final effector cell, one of the most important final effector cells in controlling toxoplasma. The other arm of this is shown over here. So IL-12 can also act on natural killer cells and do very similar things. So natural killer cells will then produce interferon gamma, which again acts on macrophages, causes macrophages to become activated, and once those macrophages are activated, they can kill the parasites. So you can kind of see this in experimental models. So we use mice as our experimental model. And so mice have been created that lack the ability to make IL-12. Their IL-12 gene has been deleted, it's been knocked out. If you infect these mice, IL-12 knockout mice with toxoplasma, they're completely unable to control the infection. They die within about a week of infection. And likewise, if you can also take interferon gamma knockout mice that cannot make interferon gamma. If you infect those mice, again, the parasite you cannot control the parasite. The mice die from overt parasitemia, or uncontrolled parasite replication. Really saying that these cytokines are key in controlling the parasite and allowing the host and the parasite to survive. So this is something we refer to as a pro-inflammatory signaling cascade or pro-inflammatory cytokine cascade. Each of these are pro-inflammatory cytokines. If these cytokines are produced in an uncontrolled manner, that can result in pro-inflammatory cytokine pathology. So these cytokines have to be regulated. If you overproduce them, they cause tissue damage, and the tissue damage can actually be lethal. Septic shock, if a septic shock caused by bacteria, it's a lethal thing that kills, I think, over 100,000 people per year in this country. And that's caused by overproduction of these inflammatory cytokines. So it's really important to regulate production of these cytokines. And another cytokine is important in downregulating these responses, preventing them from spiraling out of control. And that cytokine is here, the cytokine interleukin 10. So interleukin 10 acts on these cells and downregulates these cytokines so that they do not cause host pathology, and yet at the same time you have enough of these cytokines produced to be able to control the infection. And IL-10 itself, interestingly, comes from these same kinds of cells. T cells and natural killer cells also make IL-10. So IL-10 is an important downregulatory or anti-inflammatory cytokine. So this is what I want to really focus on. This is what a lot of work has focused on in my lab at Cornell before I came here, and also now at UNM. And so we are interested in this initial interaction between parasites and cells of innate immunity like dendritic cells that are really important in making this initial source of IL-12, acting as a source of early IL-12. So I guess about, when was it? The late 1990s, toll-like receptors were discovered in mammals. And toll-like receptors are drawn schematically here. This is just from an introductory immunology textbook. Toll-like receptors are these things here. They make these heterodimeric molecules, sometimes homodimeric molecules, and they're embedded within membranes. And then the toll-like receptors signal innate immune cells. They cause innate immune cells to produce cytokines like IL-12. And they do it through an adapter molecule called MYD88, which is shown here. So MYD88 is actually what transmits signals from these toll-like receptors to the inside of the cell. They're called toll-like receptors because there's a toll molecule expressed by Drosophila. This is where toll was originally described. It's actually a really interesting story. The toll molecule in Drosophila was described a long time ago now. I'm not even sure when, maybe early 90s, probably even before that actually, probably the 80s. Anyway, the toll receptors in Drosophila are involved in development. They're involved in establishing dorsoventral polarity during embryogenesis. But it turns out that the toll-like receptors that was discovered later in Drosophila, these toll-like receptors are re-expressed in adult flies. And in that situation, the toll receptors are involved in host defense. So it's kind of like a signaling pathway that's used in development. It seems to work pretty well in development, so they're used again in a different context in terms of inducing signaling responses that are important in host cell defense. So in the late 1990s, it was discovered, actually by this guy here, Charlie Janeway, he's a famous immunologist, and he discovered that vertebrates express genes that are related to the toll receptors. So we call them toll-like receptors. And so for us, in vertebrates, toll-like receptors are involved in host cell defense. They're involved in innate defense. And so it is an innate system of recognizing infections. So as I said, so here are the toll-like receptors. They have different specificities. So in humans, I think there are 13 different toll-like receptors. They have different specificities. They can recognize things like bacterial LPS, microbial nucleic acids. They can recognize DNA, RNA. They are also can recognize, there are some that recognize bacterial flagellin. So it's a spectrum of different pathogenic molecules that are recognized by these toll-like receptors. So these were originally described, as I said, in the late 1990s, and people were studying them in the context of recognition of bacterial infection. And so pretty soon after they were discovered and this basic signaling pathway was discovered, people identified MYD88 and they deleted MYD88 from the genome of mice so that they could see the responses of mice lacking MYD88 and how they were able to resist infection. As I said, most of this was done in the context of bacterial infection. But those of us working on other pathogens, like protozoan pathogens, were really interested in knowing whether toll-like receptors were involved in defense against eukaryotic pathogens. So this is an old study that I was involved with, actually. This was a collaboration. I actually collaborated with my old PI at NIH. And so this is actually the first study that describes the responses of these mice to a protozoan infection. And so what you can see here, here's a survival curve, percent survival, days after infection. If we infect wild-type mice with parasites, they can survive. But in mice that don't express MYD88, the infection is completely lethal. They die within 2 weeks of infection. And they die with kinetics very similar to mice that cannot make IL-12. So this was the first evidence that toll-like receptors in MYD88 were involved in host defense against a eukaryotic pathogen. The toll-like receptor specificity was more widespread than just involving recognition of bacterial pathogens. It was involved in defense against more complex organisms, eukaryotic organisms. If you look in these mice, in the days before they die, if you look in the peritoneal cavity of these mice, this is percent infected cells in the peritoneal cavity, you can see that wild-type mice have controlled the infection to undetectable levels, but in the absence of MYD88, and for that matter in the absence of IL-12, we have a lot of infected cells. So these mice are not controlling the infection. If you look in the serum of these mice, you see wild-type mice making lots of IL-12, but production of IL-12 in the absence of MYD88 and IL-12 is vastly decreased to, in some cases, non-detectable levels. So really saying that MYD88 is an important host defense molecule in resisting this pathogen. So if you kind of like fast forward about 10 years, this is kind of the situation we have now. I didn't want to go through the data that allowed us to arrive at this model because I was only peripherally involved in it. But what we now know is that toxoplasma expresses a protein called profilin. This is actually a protein that's involved in invasion. And the profilin molecule is recognized by toll-like receptors 11 and 12. And so by recognizing profilin through TLR 11 and 12 and signaling through MYD88, cells like dendritic cells receive the signal to make IL-12. So this is thought to be an important mechanism of triggering immunity in mouse models of infection. Okay, but there's a problem here. There's a complication here. And it's kind of like made us pretty uncomfortable when we started to realize that. And the complication, it's not really a complication, but the thing that made us kind of uneasy, was that it turns out that toll-like receptor 11 and 12 are actually kind of sporadically expressed in the animal kingdom. That's what's shown in this table here. So green means TLR 11 and 12 are present, red means it's absent, and red means that the TLR in question is a pseudogene and it's not expressed. So what you can see here, here's TLR 11 and TLR 12, is that there's a lot of red here and here. There are a lot of species that don't express these toll-like receptors. Mice do, obviously. We did our experiments in mice. Mice are up here somewhere. Where is it somewhere? Here we are, no, right here. Mice express it. But, for example, in primates, these receptors are absent, and that includes humans. So we do not express TLR 11 or TLR 12. And yet, I already told you, there's a lot of you are infected with toxoplasma. So you were able to survive infection even though you don't express TLR 11 and 12. So that was really disturbing to us because we were going to the NIH to get money to study toxoplasma because we said, oh, it's a serious human infection. We've got to understand how it's recognized. We're doing all these studies in mice and look, TLR 11 and 12 are involved in recognition. But it turns out that humans don't express these toll-like receptors. Okay, so somehow we're able to we're able to survive infection without these major pattern recognition receptors, as they're called. And the kind of problem, it's not really a problem, but it's kind of more widespread than that. So here's a study that came out in 2008. And in this study, a population of individuals, these were children, were identified, and these children had a genetic deficiency in expression of MYD88. They do not express any MYD88. So most of their toll-like receptor signaling pathways are inactive. And the kind of conclusion of this paper is that this population of patients are actually surprisingly resistant to infection. So they were able to identify that they had an increased susceptibility to certain bacteria, pyogenic bacteria. These are bacteria that form pus. But they're surprisingly resistant to a large number of other types of pathogens, and that includes toxoplasma. So some of these patients, some of these kids had toxoplasma. So not only do you not need TLR 11 or TLR 12, but it looks like MYD88 in general may be dispensable for resistance in humans. So the kind of uncomfortable conclusion that you come to is that in mice, TLR 11 and TLR 12 and MYD88 are clearly important in resisting microbial infections. But for humans, the situation seems to be somewhat different. These receptors are not so critical in resisting infections. So there's a big question now. So if TLR 11, 12, and MYD88 are not important in resisting infections, what is important for us? And so we are working on that question right now in the lab. Okay, yeah, right, so they're not required. So we're starting to work on that question in the lab. Here's an older observation that we made. And so what we were able to discover here, this is kind of an unexpected discovery, was that if you take MYD88 knockout mice, and if you inoculate them with a non-virulent strain of toxoplasma, so it does not persist in the host, it doesn't cause disease, this attenuated toxoplasma strain is actually able to stimulate strong protective immunity in the absence of the MYD88. So, and that's shown in this survival curve here. So these are non-vaccinated, non-inoculated mice. If you give them a virulent challenge, they die very rapidly after infection. And now if we inoculate mice, either wild-type mice or MYD88 knockout mice, with an attenuated strain of parasite, these mice are 100% resistant to challenge infection. So somehow, in the absence of MYD88, these mice have been able to generate a protective immune response. You can see that here, the level of cytokine production. We're measuring interferon gamma here. So this is the cytokine that's involved in activating macrophages. You've got to make it if you want to survive toxoplasma infection. And in this experiment, we took mesenteric lymph nodes from wild-type and knockout mice, and we cultured them in vitro. So you can see if we take cells 4 days after infection, there is indeed a defect in production of interferon gamma in the MYD88 knockouts compared to the wild-type mice. I should mention here what we do in these experiments is we stimulate in vitro with something that we call STAG, which stands for soluble toxoplasma antigen. It's basically just ground up parasites. You sonicate parasites, add them to the lymph nodes, and then measure cytokine production. So lymph node cells from day 4 infected mice are defective in interferon gamma production if the lymph nodes are derived from a knockout mouse. But look what happens 3 days later if we do the same thing. By 3 days later, the cells from knockout mice are perfectly capable of making lots and lots of interferon gamma. And again, that's consistent with the ability of these mice to generate protective immunity when they're vaccinated with an attenuated parasite strain. Right, so that brings me to UNM. And so someone in my lab, Heather Mercer is a graduate student in my lab. She is a second-year graduate student in the lab. And so she is looking into this MYD88 independent immune response in toxoplasma. So this is preliminary data, it's not published yet. But what you can see in this experiment here is what Heather did in this experiment was she inoculated mice. She gave them an IP infection. And then I believe it was 72 hours later, patient's nodding good, 72 hours later, she took the cells from the peritoneal cavity. She put them into culture, and then at 1 to 3 days after culture, she collected the supernatants and measured IL-12. And so her result is here. So what you can see is, as expected, cells from wild-type mice make lots of interleukin-12. But you can also detect IL-12 production from MYD88 knockout mice. So it's less, for sure it's less, but there's clearly an IL-12 response in these mice. So we want to know what cells are making this IL-12 and how is it involved in induction of resistance in these mice. How does it play into induction of adaptive immunity in the absence of IL-12? Well, one of the tools that we're using is our IL-12 reporter mice. So these are genetically generated, they're genetically engineered mice. And these mice are engineered to express a protein called yellow fluorescence protein off of the same promoter that's responsible for induction of the IL-12 gene. So essentially, any cell that's green is making IL-12. So what Heather did here was she infected mice again by IP inoculation. She collected cells, and then she threw the cells under our confocal microscope upstairs. And these are the kinds of results you can see. So, all cells that are green are making IL-12. What you can see here is just kind of immediately some of the things that we're seeing is that, so infected cells make IL-12. So these red things are tachyzoites. This is clearly an IL-12 positive cell. But there are also cells that are not infected that are also clearly making IL-12. And then there are other cells, kind of as a control, there are other cells that are not infected that are not IL-12 positive as well. So we have not really identified what these cells are yet. That is something that Heather is going to be doing hopefully soon. Another thing we want to do is we want to take these mice, these reporter mice, and this is going on right now in the basement, and we're crossing these mice with the MYD88 knockout mice so that we can generate IL-12 reporter mice on an MYD88 knockout background, and really get at the issue of what cells are able to make IL-12 in the absence of MYD88 signaling. So I now want to switch gears a bit and talk about what's actually going on inside an infected cell. And this gets back to this movie that I started out with, where toxoplasma actively invades cells, dives into cells. As it's invading, it's releasing proteins contained within rope trees. And so we now know that some of these proteins that are being released by the parasites are actually being injected into the host cell, and they're influencing signaling in the host cell. And so that's another major aspect of toxoplasma infection that we're interested in studying in the lab. So we found out some time ago, actually this was an observation by my wife, Barbara Butcher, who's standing right there, sitting right there. What we found was that during infection, toxoplasma stimulated very strong activation of a signaling molecule called STAT3. So that's shown in this figure here. So we're looking at infection of macrophages now. We're looking at in vitro infection. What you can see is that if we infect cells with toxoplasma, within 2 minutes of infection, we see strong activation of the signaling molecule STAT3. So we see very rapid activation within 2 minutes of infection, and its activation is sustained for almost 24 hours after infection. And it's interesting to compare this response to a response of something like bacterial lipopolysaccharide. This is kind of a prototypic microbial molecule that's used to activate cells like macrophages. And so you can see in this case, LPS also stimulates STAT3 activation, but it takes a lot longer. So toxoplasma is actually better than our positive control in inducing STAT3 activation. I should probably mention that we measure activation by phosphorylation of STAT3. So when STAT3 is phosphorylated on a particular tyrosine residue, this causes activation of this molecule. And we can easily detect this because we have phospho-specific antibodies. So this is the Western blot. And you can see what's going on in the cells down here. Here we're looking at toxoplasma in green. STAT3, we're looking at STAT3 in red fluorescence. And so you can see in the infected cells, STAT3 is being activated, it's becoming phosphorylated, and it's translocating to the nucleus in the infected cells, but not so much in the non-infected cells. So this was kind of a really interesting observation at the time, because at the time, so this was back in 2005, people were still thinking of toxoplasma as kind of a silent infection. So something that invades a cell and creates a vacuole, and something that, by virtue of the fact that it's in a vacuole, it is sequestered from the host cell, and it's not really, the host cell is not really responding to it. So this is actually, I think it's really the first evidence that really shows that the host cells are actually being manipulated by the parasite. They're undergoing signaling responses in response to infection. So we and several other groups in the field were really interested in this response. And so it turns out that a parasite protein called ROP16 is a protein that's involved in activating STAT3. ROP16 is one of these rotary molecules. It's expressed within the rotaries of the parasite. And during invasion, this molecule, along with other rotary proteins, is released from the rotaries. The rotaries release their contents. And ROP16 is injected into the host cell cytoplasm, and in the host cell cytoplasm, it activates STAT3. So ROP16 is a host-directed kinase. And again, this was an amazing sort of discovery in the field, that this eukaryotic pathogen was expressing kinases that were actually directed to the host cell. So this was actually a really exciting time in the field when these discoveries came out. So there was a nice Nature paper that came out by a group from Stanford, and in this paper what they showed was that if you tag ROP16 with an HA tag, you can follow it in a cell. Here are the cells up here. Here are parasites up here. Same cells down here. So the parasites are green, they have ROP16. But look at this, the host cell nucleus has ROP16. So ROP16 is being released by the parasites, and it's finding its way into the host cell nucleus. And in fact, ROP16 has a classical nuclear localization sequence. So over here, we kind of formally demonstrated that ROP16 was involved in STAT3 activation. So this is a Western blot looking at phosphostat3. And so again, if you infect macrophages with wild-type parasites, we see strong STAT3 activation. This is phosphorylation of STAT3. This is due to phosphorylation. It's not due to increased synthesis of STAT3. Here are total levels of STAT3, and they remain pretty much the same all the way across. So it's really triggering phosphorylation of STAT3. If we knock out ROP16 from parasites, this was done by a collaborator in Dartmouth, David Bezick, who I know some of you heard his seminar. He came out and visited last year and gave a nice seminar. He created ROP16 deletion mutants, and then he was nice enough to send them to us. And so what you can see if you infect macrophages with the ROP16 knockouts is that the STAT3 phosphorylation response is maybe an initial response, but it's not maintained. If you take these same parasites, these knockout parasites, and you make a complementation mutant, so you reinsert ROP16 into these mutants, you restore the ability of these parasites to phosphorylate STAT3. Okay. So this was really demonstrating that ROP16 was the molecule that's involved in activating STAT3. This is the kinase that's involved in phosphorylating STAT3. So I should tell you about the STAT3 activation pathway. It's a pathway that's used by a lot of cytokines. And so this is how it works. So STAT3 is actually involved in signaling mediated by IL-10. And remember I told you right at the beginning that IL-10 is an important anti-inflammatory cytokine. It downregulates inflammatory responses. The way it works is that it's recognized by an IL-10 receptor, which is a dimeric molecule. Binding of IL-10 to the receptor brings these two subunits together. And these subunits have cytoplasmic molecules called JAK molecules, which stands for Janus kinase. These are tyrosine. These are molecules involved in tyrosine phosphorylation. These molecules are brought into close proximity, and they transphosphorylate. So the JAK molecules phosphorylate each other. They tyrosine phosphorylate each other. Once these molecules are phosphorylated, STAT3 is recruited to the receptor, and it is also phosphorylated by these JAK molecules. When it's phosphorylated, it dissociates from the receptor, and it dimerizes right here. And once it's dimerized, it then translocates to the host nucleus, and it binds to target promoter regions involved in anti-inflammatory gene transcription. So STAT3 is a signaling molecule that's involved in mediating the effects of IL-10. So what it looks like, what everyone agrees on now, is that toxoplasma is hijacking this pathway. It's using this pathway. Toxoplasma, when it invades, is injecting this protein, ROP16, and ROP16 is a kinase that directly phosphorylates STAT3. It's bypassing this whole initial steps in signal transduction. So what are the biological consequences of ROP16-mediated STAT3 activation? And we don't really completely understand it yet, but here's one experiment that's informative. What we're looking at here is production of IL-12 by macrophages. This is an in vitro production of IL-12. So what you can see is if you take macrophages and infect them with wild-type parasites, RH is just a strain of parasite that we use, and these are wild-type parasites. So we can see some IL-12, but certainly not really a lot over the median controls. If you take these parasites and delete ROP16, now these parasites can stimulate production of a lot more IL-12. And if we put ROP16 back into these parasites, we decrease the response back to wild-type levels. So one thing that ROP16 seems to be doing is down-regulating production of IL-12 in infected cells. Oops. But it's a complicated molecule, and that's why I was saying we don't really understand what it's doing yet. And the reason it's complicated is because ROP16 is kind of promiscuous, so it can activate other STAT molecules. So we also found that ROP16 activates STAT6. So this is a completely different signaling molecule involved in other cytokine responses. And so you can see really pretty clearly, here is the deletion mutant, the parasite deletion mutant. And if we infect macrophages with the deletion mutant, we don't see any activation of STAT6. And then if we put ROP16 back into these parasites, now we see activation of STAT6. And we see induction of a lot of STAT6 associated proteins like arginase 1. So we've kind of got to deal with now when we're trying to figure out what what ROP16 is doing, we've got to think about STAT3, but we've also got to think about STAT6 now. Completely different transcription factor. Similar name, 2 times 3 is 6, but they're completely different. They're completely different molecules. Really muddies up the situation. And so we're continuing to look at STAT activation in the lab now. And so this is some work that an IMSD student in the lab, Huang Gui, aka Ray, is looking at this. And what Ray has discovered is that this molecule here, here's another STAT molecule, is also activated by toxoplasma. So now we have STAT5 as another STAT that's activated by toxoplasma. So in this experiment, Ray infected dendritic cells with increasing amounts of parasites. And so you can see there's a nice STAT5 activation response. This is looking at different times after the in vitro infection. So another STAT molecule is activated by toxoplasma. What I will say about this data is that I think it's pretty clear, just looking at this, that activation of STAT5 is not going to be the same as activation of STAT3 and STAT6. The reason I say that is because if you look at total levels of STAT5, in non-infected cells there's no STAT5, and STAT5 itself is being induced, the protein is being induced by infection. That's not the case for STAT3 and STAT6, which are molecules that are present in non-infected cells, and what is happening in those situations is that STAT3 and STAT6 are being phosphorylated. In the case of STAT5, it seems that toxoplasma is phosphorylating these molecules, but it's also upregulating expression of these molecules. So Ray is interested in kind of chasing this down and asking more about the biology of this effect. So kind of a summary slide for ROP16. So we think there's at least 2 roles for ROP16. I think there's probably more actually. But ROP16 is a molecule that is injected into the host cell cytoplasm during invasion. It can phosphorylate STAT3 and STAT6. These molecules are going to the nucleus. And at least for the case of STAT3, we think this is involved in IL-12 downregulation. So as I said, very possibly there are other targets of ROP16 that we don't understand yet. And that's something that I'm interested in pursuing in the near future in the lab. The last thing I want to talk about is some new data that I'm actually really excited about this, and what we are interested in looking at is the role of long non-coding RNAs during toxoplasma infection. So this kind of like goes back to when the human genome was first sequenced. I think it's in 2000 when the Nature paper came out. The human genome was sequenced, and the kind of major surprise from the data that came out of that was that humans only have on the order of, I think it's around 20,000 genes. Everyone was really surprised by the fact that you could make a human out of only 20,000 genes. It's not a big number, it's a small number. And so the people were really puzzled by that. So there's only 20,000 genes, so what does the rest of the genome do? That only accounts for about 3% of the genome, I think. And so for a number of years, people kind of thought that the rest of the genome was just junk. Kind of junk that we carried around with us. But it's now becoming clear that a lot of that genome is actually functional, and it's actually expressed, and one class of molecules that are expressed are called long non-coding RNAs. So these are RNA molecules. They are not translated into proteins. They're by definition over 200 nucleotides long. And so there's a lot of research going on the role of long non-coding RNAs, and there's a lot that's not known about them. So one thing I really like about this project is really nothing is known about long non-coding RNAs, and that's particularly true for their role in infection. And so that's kind of like my favorite situation, is where you're going into undiscovered country and you don't really know what you're going to discover, and I really find that exciting. But long non-coding RNAs actually have a lot of different functions, and so some of their functions are summarized in this review figure here. It just came out very recently. So long non-coding RNAs have a lot of different functions, And again, we're just still trying to understand the spectrum of different functions that they have. But for example, some long non-coding RNAs are exported into the cytoplasm, and they can interact with proteins in the cytoplasm. In the cytoplasm, they can also interact with messenger RNA and influence expression of messenger RNA. There's actually new evidence that long non-coding RNAs can interact with microRNAs and influence the activity of microRNAs. Other long non-coding RNAs are actually retained in the nucleus, and they can bind to transcription factors and influence the activity of transcription factors, so either upregulate transcription factors or downregulate transcription factors. They can also interact directly with DNA and influence gene activity in that manner. So it's kind of like going to the zoo and all of these different species that do different things, that have different properties. It's kind of like the situation with long non-coding RNAs. We're kind of going to the zoo and discovering all of these different functions. And again, as I say, their role during infection, not just toxoplasma, but any infection, is pretty much completely unknown. There's a relatively large number of papers that provide evidence for the function of these different long non-coding RNAs during development, for example. But in the context of infection, which is what we're interested in, there's really not much known about them. So we were really interested in seeing what we could find out about long non-coding RNAs in the context of toxoplasma infection. And so the work I'm telling you about now is really spearheaded by a postdoc in my lab, Kayla Menard, who's been here almost exactly a year now. And so she's formed a link RNA team with Rhianne Haskins, who's another IMSD student. And so they're kind of really spearheading this effort to try and understand what link RNAs are doing, if anything, in the context of toxoplasma infection. So what this shows here is a microarray that we did. And this microarray, you buy it from a company, you buy it from ArrayStar if anyone's interested. These microarrays have, I believe the number is 36,000 different link RNAs that have been identified. And so we're looking at expression of these link RNAs, and in this experiment what we did was we infected macrophages with the Rh strain of the parasite. This is a virulent strain of the parasite. And then in parallel we infected macrophages with a low virulence parasite strain. And so this shows the results of that microarray. These are volcano plots. And so what these plots show, I had to learn this, what these plots show is that on this axis we're looking at fold change in expression relative to non-infected. So anything over here is being upregulated, anything over here is being downregulated. And then on this axis we're looking at the probability. So these experiments were done in biological triplicates. And so we can start to identify link RNAs that are being upregulated by toxoplasma infection and a smaller subset that are being downregulated. And we can start to make comparisons between infections with a high virulence parasite strain and a low virulence parasite strain. And that's kind of what we're doing now. We're kind of sifting through this data, we're validating it by conventional PCR, and it does, for the most part, thankfully, it does replicate the results of the array. And so we're starting to see some interesting things. And here's one interesting thing that we've noted. So just looking through the link RNAs that are upregulated by infection, one link RNA that we've identified is a link RNA that's associated with the SOX2 gene. The reason we're interested in this is because SOX2 encodes a protein called suppressor of cytokine synthesis. So this is a protein that's involved in downregulating cytokine responses. Here's how the protein maps here, and here's the link RNA up here in green. This link RNA is referred to as link 007. That's Kayla's term, she came up with that. So what you can see here is that this link RNA, 007, is upregulated during virulent infection, but it's not upregulated during the low virulence infection. And the same pattern is followed by the SOX2 RNA. It's upregulated during virulent infection, but is not upregulated in the low virulence infection. So we're interested in kind of chasing this down. It's just an association at this point. We really need to think about how we can generate cells or even mice that don't express this linked RNA and see how that affects the ability to resist toxoplasma infection. What we do know about link 007 is that it is also regulated by ROP16. That's shown in this graph here. Here's link 007. And so if we infect macrophages with wild-type parasites, ROP16 knockouts, or the complementation mutant, we can see that in the absence of ROP16, this link RNA is not being upregulated. And again, it follows the same pattern as SOX2 expression. So this is a link RNA. We don't know what it does, but we at least know the parasite protein that's involved in inducing its expression, and that protein is ROP16. Okay, so that's most of the data I want to show you. I just wanted to get back to this slide. I hope that what I've showed you kind of emphasizes this point that this parasite, Toxoplasma, is a really smart parasite, and it is really able to manipulate the immune response. It's able to be recognized in some sense, but it's also able to avoid inducing too strong of a response. It's really achieving this happy balance in between the two. And as I said, I think the data that's coming out of our lab is really emphasizing this point. I know I'm running late. I just wanted to say, in the last point, it would be remiss of me not to mention the other people in my lab. I just wanted to mention what they're doing. Obviously, I didn't have time to talk about what they're doing. But Cameron Rankin is a first-year graduate student in the lab. He started as a technician in the lab. And he has discovered that if you block post-cell beta-catenin, this can also block infection. So this is something that he is studying in the lab. You can ask him about it because I've run out of time. But this is what he's been working on for about the last year. Alicia Romero is an IMSD student, and she's developed this really nice in vitro technique for inducing differentiation of parasites into the cyst-forming stage. So she can infect neurons in culture, and she can get the parasites to form cysts in culture. And so she is looking at the different factors that influence cyst formation. Okay, acknowledgements. I'll just quickly. So this is my lab right now. Nice, good lab, nice productive lab. We're generating this data. A lot of this work was done when I was at Cornell by various graduate students and postdocs and technicians. And as I said, the knockout parasites were generated by David Bazik, working at Dartmouth Medical School. My funding has all come from the National Institutes of Health, and since I've been at UNM, SETI has also provided financial assistance. And that's it. So this is the UNM Toxo team. Thank you. Yes, Chris.
I have a couple questions about the ROP16.
Yeah.
Do the toxo cells themselves express stats or other substrates?
Yeah, right.
ROP16
Good question.
all just as an interactor with the host cells. And secondly,
Right.
is there anything known structurally about it? Is it perhaps homologous to the JAK kinase?
I don't think it's homologous, no. And so the first question, no, toxo does not seem to express stat proteins. Eukaryotic parasites in general don't express tyrosine kinases. So what people think is that this is actually ROP16 originated as a serine threonine kinase that somehow acquired the ability to tyrosine phosphorylate host substrates. That's the kind of latest thinking. So parasites, no, they don't express stat proteins. Q.
Yeah, another question on ROP16. It seems to be the switched arming at the parasite is very versatile. You actually have a number of stat factors in the host cells.
Right.
Are there indications from characterization of these proteins that ROP16 is polymorphic? Can it be modified itself to meet these different activities? And would that then be interaction with the host cell, or the parasite itself potentially generate a sort of differently modified ROP16?
Yeah. Okay. So that could be a long answer. So ROP16. So we know that ROP16 can activate STAT3, STAT6. But I mean, this is something that we're looking at in the lab. I don't know. Maybe some of these are off-target effects, and so maybe we don't really understand what the true target is. For that matter, we may actually not even be looking in the right cells. Most of our experiments are done in macrophages and dendritic cells, but toxoplasma infects all kinds of different cells. Maybe we're not looking in the right cell. And you know, it gets worse than that because maybe we're not actually looking in the right host. Everything we've done is in mice, but the cat, for example, is important for the life cycle of toxoplasma. And so maybe there are ROP16 effects that have yet to be discovered in other host cell, different species of host cells. And as to your other question, yes, ROP16 is polymorphic, and I didn't get into that because I didn't want to. But yes, there are different strains of toxoplasma that do not express active ROP16, and that's actually how ROP16 was identified, was by crossing parasite strains that either express or do not express ROP16, and actually chasing down virulence loci, and that's how ROP16 was identified. Yeah, you're in it. Ready?
Any evidence of your link RNA 007 in
007.
007 in human?
In
you
humans?
just look at certain,
Right.
certain samples
Yeah.
from humans with and without, and you find a link.
Yeah, okay, so Kayla can chip in if she wants, but it's not a conserved link RNA.
Ah.
So there's no direct connection with in humans. We are actually in that array we did
You guys should do the exact same experiment, but with human macrophages.
with human macro, yeah.
Because then you go to an
That's
IH
right.
directly. Like
Yeah.
that's-
So one thing that we had, one thing that I didn't talk about is that there are some of those link RNAs that are upregulated are classified as ultra-conserved link RNAs that are in humans. I was just interested in this particular one because it
Well,
is
the SOX
associated with
link is
SOX2,
really-
exactly.
yeah,
Yeah, so, yeah. Michelle.
Lots of questions, so I'll just ask
Okay.
two, which may or may not be. So, this crazy vacuole, I forgot the name of it now,
Parasitophorus.
parasitophorus,
Yeah.
that things enclosed in. Are the toll receptors that are on that membrane originally on the plasma membrane and they just kind of get swept up? Or are they later transported there and then it gains some sort of its own membrane identity within?
No, I think you're overthinking it. So the toll-like receptor, so the
I guess
parasite...
my question is, does that membrane have some kind of identity?
Okay, so here's what let me answer. So the parasite is harbored within a parasitophorus vacuole. Membrane that vacuole, there's no evidence that vacuole recruits toll-like receptors.
Oh.
So in fact,
So how is the profilin being?
so the thought is, well actually we don't really understand, but one thought is that actually it's non-infected cells that are actually responding. And so maybe it's dead parasites that will be phagocytosed and they would interact with the toll-like receptors.
Okay. On your diagram, you're drawing that.
Pardon?
On your cartoon, you're drawing that.
That was, yeah, so that was a cell that had phagocytosed.
Okay.
Okay, yeah. Not clear, yeah, I agree.
And so then my other question is, you have these two independent pathways, and the responses for your interleukin 12 and your MyD88, you're looking at days. And then with the STAT receptors, you're looking at minutes, but they're both giving you this cytokine response. And I was wondering how those two pathways interface with each other, and if is one required for the other, or are they at all connected?
Well, I think they are connected because one piece of data I showed you with IL-12 production, when you delete ROP16, IL-12 goes way up. So the IL-12, you're right, is measured in the context of days after infection. Our signaling responses are within minutes of infection. So I think that the signaling kind of takes a while to kind of translate to protein production and secretion to reach levels that you can actually detect, and all of that takes time.
Any other questions? Okay, thank you. Aaron.